Hello, dear students. I welcome you all on behalf of Sri Jendra Rank Zone classes. Dear student, we are in a continuation of the chapter that is Principle of Inheritance and Variation. And in that chapter, we are going to continue today one more small topic that is chromosomal theory of inheritance. If we talk about this theory, in this we have to note down some of the theoretical points and we have to understand that one by one. So we will be enlisting that points here one by one. Okay, so let's talk about this in detail. If we talk about this, then, dear students, this chromosomal theory of inheritance was proposed independently by Sutton and Boveri. So it was the two scientists who put this theory forward. And these two workers, they found close similarity between transmission of hereditary trait, transmission of hereditary traits and behavior of chromosome. Transmission of hereditary trait and behavior of chromosome. While passing from while passing from one generation to the next, to the next through the agency of gamete. तो ये इन सटर एंड बोवरी ने दी, इन्होंने ये सिमिलैरिटी ऑब्जर्व की कि जब एक जनरेशन से दूसरी जनरेशन जब कोई हेरिडिटरी ट्रेट या क्रोमोसोम का इनहेरिटेंस होता है, ट्रांसमिशन होता है, तो वो क्लोज सिमिलैरिटी में है, वो एक जैसा है, ऐसा इन्होंने ऑब्जर्व किया। और ये जो सारा नॉलेज था वो सटन ने यूनाइट किया। सो सटन united united the knowledge of chromosomal segregation with Mendelian principles. And call it chromosomal theory of inheritance. तो यहाँ पे सटन ने इंपॉर्टेंट रोल प्ले किया। इन्होंने पूरा नॉलेज यूनाइट किया, क्रोमोसोमल सेग्रीगेशन कैसे हो रहा है वो, और मेंडेलियन प्रिंसिपल कैसे वर्क कर रहा है वो, और उनको यूनाइट करके उन्होंने ये कुछ पर्टिकुलर क्रोमोसोमल थियरी ऑफ इनहेरिटेंस यहाँ पे प्रपोज की। नाउ इस पूरी थियरी जो रखी गई उसके कुछ साइलेंट फीचर्स है। सो नाउ वी आर गोइंग टू टॉक अबाउट some of the salient features salient feature of chromosomal theory of inheritance. And the salient features of this chromosomal theory of inheritance are as follows. I think that's the first feature is that like the like the hereditary traits, the chromosome retain their number, structure, and individuality throughout the life of an organism. Means here the similar to the hereditary trait, which are the variant forms of the character, the chromosome also retain the number, structure, and individuality throughout the life of an organism and from generation to generation as well. Here, the two neither get lost nor mixed up. Means what? Trait as well as chromosome. They behave as units. Means we can say these two chromosomes are not lost or not mixed up. They behave as unit. Okay, so this is the first thing which shows that the retainance of their characteristic feature like number, structure, and individuality throughout the organism and from generation to generation, that helps us to understand this whole particular thing. Now, apart from this, the second thing which we will discuss in the salient features of chromosomal theory, that both chromosome as well as genes occur in pair. It is seen that both chromosome as well as genes occur in pair in somatic or diploid cells. Chromosomes are also in pair and genes are also in pair. Two alleles of gene pair, two alleles of a gene are located on homologous sites on homologous chromosome. So here both chromosome as well as we can say both genes which are found in a pair in somatic as well as diploid cells and the two alleles of a gene are located in homologous sites on the homologous chromosome. So these are the two alleles. Now these both chromosome occur in a pair that is seen here. Now after this it is seen that both chromosome as well as gene both chromosome as well as genes segregate at the time of gamete formation such that only one of each pair only one of each pair is transmitted to a gamete. The fourth one is gamete contain only one chromosome. Gamete contain only one chromosome of a type and only one of the two alleles of a trait. And the fifth one is the paired condition of both chromosome as well as Mendelian factor is stored during fertilization. So both through when the gamete fusion occur, this both chromosome as well as Mendelian factor or a gene come in a pair by the process of fertilization. So this all things showing a what a similarity between the chromosome and genes. Thus homologous chromosome synapse or make a pair during meiosis and then separate or segregate independently into different cells, which establishes the the quantitative basis for segregation for segregation and independent assortment of hereditary factors. Okay, so this all points, if you compare, then they will be showing the similarity in the inheritance of chromosome and genes. So because of this, Sutton and Mori concluded that these genes are nothing but presence on the what? A chromosome. So ultimately chromosome are getting inherited, and this is known as chromosome theory of inheritance. Now further, the experimental verification of this chromosomal theory of inheritance was done by Thomas and Morgan, which we will discuss in the linkage and recombination part. But here there is a one question in NCRT that we need to discuss, which will help to differentiate between gene and chromosome. And the question is that there is a two columns are given, and we have to identify that which column is for the gene and which column is for the chromosome. So question is, can you tell which of this column which of this column A or B represent the chromosome and which represent gene? Now if we talk about column A, which is given here, in column B, if we note down the points, then in column A it is given that they occur in pair, and in column B also it is written that occur in pair, so no difference. In second point they have written that they segregate at the time of gamete formation such that only one of each pair only one of each pair is transmitted to a gamete. And here they have written that segregate at the time of gamete formation and only one of the each pair is transmitted to a gamete. Means here also you will not see the difference. But there is a difference in the just only a third statement, and here they have written that independent pairs segregate independently of each other. And in column B, one pair segregate independently of another pair. Okay, so here the two statements are similar one, but the third statement there is a only difference is that in the column A they have written that independent pair means any pair. If they segregate independent of each other. Means if I compare a first pair with the third pair, or third pair with the fourth pair, or fourth pair with the 10th pair, or 10th and 7th pair, or 8th pair, any pair, if I compare randomly, the inheritance is not going to be particularly dependent on each other, because they are the separate entities, separate bodies. Okay, can be if there is a separate bodies, then it is possible that independent pairs segregate independent of each other. But if we talk about a one pair segregate independent of another pair, means we have to locate two pair here only. We can't say independent, or randomly we can't compare the many things at a time. Here we have to select those two things, and for those two things only, if we see, if we can say that one pair is segregate of independent of another pair, then this statement will be applicable to this. Okay. Now, why like this? So, if we talk about the in column A, independence randomly independence can occur in case of chromosome. Okay, in case of gene cannot occur, because gene can be present on the same chromosome. If they are present on the same chromosome, then the dependence will occur for one pair of chromosome to the another pair of chromosome. So, linkage can be established in case of gene. At that time the independent pairs segregate independent of each other will not be possible. So column A is representing chromosome and column B is representing gene. Okay, so let's answer this, and answer is saying that column A is chromosome and column B is gene. Okay, so column A is chromosome and column B is gene, because chromosome can only show the independent pair in segregation independent of another pair anyhow. But for a gene, we can't say that independent pairs segregate independent of each other, because we have to locate the gene. If we use independent term and if we locate the two gene on the same chromosome, then the statement will be false. So we have to locate the two gene. If these two gene are present on the non-homologous chromosome, then it is possible that the independent segregation occurs. But if they are present on the same chromosome and linkage is getting established, then at that time we can't say the independent pair segregating independent of each other. So one pair segregate independent of another pair in case of gene when they are present on the non-homologous chromosome. Okay, so that is about the what we can say this gene and pair, and that is all about the chromosomal theory of inheritance. Now its experimental verification we will discuss in the next lecture, where we will talk about the linkage and recombination. Okay, till that, thank you.